Hello everyone, welcome to my channel Physics for Engineers. In this video, we will be going to discuss about the advanced characterization technique for materials characterization. One of the most advanced characterization technique is scanning electron microscopy. Scanning electron microscopy is commonly known as SEM. In this video, we will explore one of the most powerful characterization technique used in material science, nanotechnology, physics, chemistry, and engineering research. Before discussing the technical aspect, let us first understand why SEM is so important in materials research. When we study the materials at the nanoscale, the conventional optical microscopes became insufficient because of their limited resolution. SEM overcomes this limitation by using a focused beam of electrons instead of a visible light. Since electrons process much shorter wavelengths than the visible light, SEM can produce high detailed images with exceptional resolution. As shown in this slide, the SEM offers several advantages. The first one is it provides the high resolution images of surface morphology. This means we can visualize the surface structure of the materials, including roughness, pores, cracks, particles, and nanostructures. The second one is the SEM enables elemental and composition analysis through the techniques such as energy dispersive X-ray spectroscopy, formerly known as EDX or EDS. This allows researchers to determine which elements are present in the sample. The third advantage is SEM helps to study the microstructure and shape control. The researchers working in nanoparticles or nanorods, nanotubes, thin films, and composites, they frequently use SEM to evaluate the morphology and to optimize the synthesis conditions. You can also see the examples of various nanostructures on the right side of this slide. These images demonstrates a variety of SEM observed in different materials architecture, ranging from spherical particles to the porous network and nanowires. These kind of images we can obtain using this scanning electron microscope. The schematic at the bottom, right, you can see the illustration how the incident beam of electrons interact with the material and generates the signal such as like the secondary electrons and backscattered electrons and X-rays. what is the importance of each kind of electron beams. So here you can see clearly the incident beam of electrons is splitting into three portions: secondary electrons, backscattered electrons, and the X-rays. These signals provides the valuable information about the surface morphology, composition, and crystal structure. In modern nanotechnology research, SEM has become an important tool because it bridges the gap between the materials synthesis and the materials performance. Because the materials performance depends highly on the morphology, size of the material. The size and morphology can be controlled during the synthesis. So, the characterization is the bridge between the synthesis and the performance of the material. Like we can say it is a bridge between the uh, synthesis and application. Whether we are developing a photocatalyst or a battery material or a supercapacitor or a sensor or any biomedical material, for any kind of material, the SEM helps to understand how the structural influences the properties, how the morphology affects the performance. That we can understand from the SEM. In this video, we will learn the fundamentals of electron matter interaction, the SEM instrumentation, imaging modes, sample preparation techniques, the EDS analysis, and advanced applications in material science and nanomaterials research. In order to understand SEM, we must first understand what happens when an electron beam strikes the material surface. When a highly focused beam of electrons is directly incident on the surface or the specimen, numerous interactions occurs between the incident electrons and the atoms present in the sample. These interactions takes place within the region called the interaction volume. You can see the left side of the slide. These incoming electrons transfer the energy to the atoms of the specimen through elastic and inelastic scattering processes. The elastic scattering occurs when the electrons changes their direction without significant energy loss. These interactions primarily generates the backscattered electrons, which provides the information about the atomic number contrast. The inelastic scattering occurs when the electrons lose energy while interacting with the sample atoms. These interactions generates several signals, including secondary electrons, characteristic X-rays, and Auger electrons. Let us discuss these signals individually. You can see the first signal, secondary electrons, which provides the information about the surface topology. These secondary electrons are the low energy electrons emitted from the near surface region of the sample. Almost it is reflecting back from the upper surface of the sample. And it is due to the inelastic scattering, so it is losing the energy. That's why these scattering, these secondary electrons are the low energy electrons, because they originates from the surface. They provides excellent topographic information and produces the high resolution surface images. The second signal is backscattered electrons originating from the elastic scattering. Because of this, these backscattered electrons are having high energy and reflected back from the deeper region of the sample. Since the heavier elemental scatter electrons more effectively, the backscattered electrons imaging provides the compositional contrast. The third signal is characteristic X-rays, which are generated when inner cell electrons are ejected and the vacancies are filled by the high energy electrons. These X-rays are unique to each element and forms the basics of energy dispersive X-ray spectrometer, that is EDS analysis. So we can get the elemental information of a sample, like what kind of elements present in the sample and what is their atomic percent, what is their weight percent in the sample. That information we can get from the characteristic X-rays. The another important signal is Auger electrons, which are useful for the surface chemistry studies. So we can also highlight the effect of accelerating voltage. At low voltage, the electrons penetration, like the penetration depth of electrons is very small, resulting in high surface sensitivity. At higher voltage, the electrons penetrates deeper into the specimen and generates more signals from the bulk materials. So understanding electron matter interaction is essential because of every SEM image and every elemental analysis result originates from these interactions. The quality of information we obtained depends on how electrons interact with the specimen and which signals are collected by the detectors. These fundamental principles forms the scientific foundation of all SEM imaging and analytical techniques. Let us now discuss the major components of a scanning electron microscope and understand its working principle. A SEM instrument consists of several important subsystems that work together to generate high resolution images. At the top of the instrument is the electron gun, which serves as the source of electrons. It generates a beam of electron that will eventually be focused on the specimen. The electron beam then passes through the condensed lens. These lenses control the diameter and intensity of the beam. By adjusting these lenses, researchers can optimize the image quality and resolution. Next, the beam passes through the aperture, which helps remove unwanted electrons and improves the beam coherence. The beam is then focused by the objective lens to form a very fine probe on the specimen surface. The diameter of this probe largely determines the achievable resolution. Then the sample is placed inside the sample chamber, which is maintained under high vacuum conditions. The vacuum is essential because electrons can be scattered by air molecules, leading to image degradation. To maintain the high quality imaging process, the vacuum in the sample chamber is very much important. The SEM also contains several detectors, including secondary electron detector, backscattered electron detectors, the high energy X-ray detectors. These detectors collect the signals generated from electron matter interactions. Now, let us understand the working process step by step. You can see here, the first process is the generation of electrons from the electron gun. Second, the beam is accelerating to a high energies. Then the third step, the electromagnetic lens focus the beam into a fine probe. The fourth step, scanning coils raster the beam across the sample surface. As the beam scans point by point, the signals are generated from each location by the multiple interactions between the electron beam and the surface. These signals are detected and converted into the electrical signals. Finally, a computer processor the signals and reconstructs an image, and that will be displayed on screen. So we can store the images and we will get the morphology of the sample specimens. So here it's also introduce the different types of SEM systems. The conventional SEM operates under high vacuum, and the field emission scanning electron microscope, formerly known as FESEM, uses a field emission electron source and provides significantly higher resolution than the conventional SEM. The third one is environmental SEM that allows us to generate the images of the samples under controlled gaseous environment. These are the three different kinds of scanning electron microscopes have been using in materials research. So one important question is why vacuum matters. Vacuum prevents electron scattering, that improves the beam stability, it enhances the resolution and increases the detector sensitivity. In summary, the SEM is the sophisticated instrument that combines electron optics, electromagnetic focusing, scanning systems, detectors, and computer processing to create highly detailed images of material surface. Understanding the instrumentation is crucial because every image parameters we adjust directly influences the final image quality and analytical results. So you can see sometimes at the bottom of the SEM image, you can see the particular instrument conditions, at what resolution it was done, what is the applied voltage, everything will be given at the bottom of the SEM image. That information is very much essential to understand the surface morphology. So in the next slide, we will begin exploring one of the most important imaging mode in the SEM. That is the secondary electron imaging, which is widely used for the surface morphology analysis. Let us now discuss about the one of the commonly used imaging mode in scanning electron microscope, that is the secondary electron imaging. In this mode, primarily used to obtain the detailed information about the surface morphology of the materials. To understand the secondary electron imaging, we must first know how secondary electrons are generated. When the primary electron beam strikes the sample surface, it transfers energy to the atoms present in the material surface. As a result, the low energy electrons are ejected from the outer shell of the atoms. These emitted electrons are called secondary electrons. An important characteristic of these secondary electrons is that they possess very low energy, typically less than 50 electron volts. Because of this low energy, only electrons generated very close to the surface can escape from the material. As you can see in the image, the escape depth is only a few nanometers. This is why the secondary electrons provides the excellent surface information. So we can get the information about the surface topology and high resolution morphology from the secondary electrons. The working principle of the secondary electrons imaging is very straightforward. A focused beam of electrons scans across the sample surface. The secondary electrons emitted from different regions of the specimen are collected by the secondary electron detector. The intensity of these collected electrons is converted into the brightness variations on the screen. So forming an image is the final output from the secondary electrons. One of the major advantages of the secondary electron imaging is its outstanding topographic contrast. Features such as an edge, corners, pores, cracks, fibers, nanoparticles and rough surfaces properly, clearly, because they emit different number of secondary electrons. Notice that the illumination showing edge enhancement. Regions with sharp edges and protrusions appears brighter. This happens because more secondary electrons can escape from these areas and reach the detector. Conversely, the flat or recessed regions emits very few detectable secondary electrons and therefore appears dark. This phenomena gives the secondary electron imaging a three-dimensional appearance, makes them extremely useful for morphological studies. In this slide, I have presented several examples of secondary electron images, including nanoparticles, spherical particles, nanorods, the porous structure, and a thin film. You can see the difference. These examples demonstrates the versatility of the secondary electron imaging in nanomaterials research. Researchers often use secondary electron images to determine the particle shape, particle size, distribution, porosity, and surface roughness. Several parameters influences the quality of secondary electron images. That is, low acceleration voltage increases the surface sensitivity. A smaller working distance generally improves the resolution. Proper beam current ensures the sufficient signal generation without damaging the sample. The conductive coatings such as gold or platinum can also improve the image quality by minimizing the charging effect. That's why before doing the sample analysis, the sample will be coated with either gold or platinum to excite the electrons more. However, some challenges are associated with the secondary electron imaging. One is non-conductive materials may accumulate electric charge during imaging. This phenomena, known as charging, can cause image distortion and brightness fluctuations. The sensitive materials may also experience beam damage if exposed to the electron beam for extended period of time. To summarize, the secondary electron imaging is the preferred SEM mode of operation for studying surface morphology because it offers high resolution images, excellent edge contrast, and detailed visualization of nanoscale surfaces with higher resolution. For many nanomaterials studies, secondary electron imaging serves as the first step to understand the material structure and morphology. Thank you for your attention. We will discuss the backscattered electrons, energy dispersed X-ray spectrophotometer, and sample preparation for SEM and other advanced applications of SEM the next video. Thank you.